Alright, so let's get to actually making two cells out of one. We're going to follow this picture through, and as you're looking at your notes, you may want to label what's happening in each one of these steps. Now, remember, this is just a picture, nice cartoon of what's happening, but it'll give you sort of a visual of the different parts of mitosis. We're going to start with prophase, sometimes also referred to as prometaphase. Here, this cell is in early prophase, and this cell is in late prophase. Let's look at what the cells are doing, and then we'll come back and look at the picture. So first, the DNA is going to coil up into visible chromosomes. When the cell's doing its job, it needs the DNA to be open so that RNA polymerase can get to it. It can make messenger RNA, create proteins, etc. But during mitosis, that whole process is going to stop and the DNA is going to tightly coil up. Next thing that's going to happen is the nuclear membrane is going to completely disappear. And finally, the centrioles are going to start to migrate to each side of the cell and grow fibers that will attach to the centromere of each chromosome and the cell membrane on each end. And they're going to form what's called the spindle fiber. So let's look at what that looks like. So here in early prophase, we start to see the chromosomes. Originally, the DNA was not tightly backed up. And we start to see little holes in our nuclear membrane form. Here's our pair of centrioles, and they're starting to move to each side of the cell and grow fibers. By the time we get to late prophase, now our nucleus is completely gone. The centrioles are migrated to each side of the cell. Fibers are attached to the outer cell membrane and to each side of each chromosome. Next we have metaphase, and metaphase is when the fibers have pulled on both sides of the chromosomes in sort of a tug of war that evens out the chromosomes in the very middle of the cell. So they're going to meet up in the middle. Metaphase, fibers pull on the chromosomes until they're aligned end to end in the middle of the cell. Now, in our pictures, this looks really pretty. At the end of this section, I'll show you a short video about what this looks like in a real cell, and you can see that they don't line up perfectly end to end, but the centromeres, the centerpiece, is at the center of the cell. Next we have anaphase, and again in our graphic here we have early anaphase and late anaphase. An anaphase is when the fibers pull sister chromatids apart. So remember, a sister chromatid is an identical side of a chromosome that's been duplicated, and during anaphase those are going to get physically separated. So we can see here the two sister chromatids have been pulled apart, and half are moving to one side of the cell, half are moving to the other. Here we're starting to see the cells pinch off. Eventually this will become two cells, and all the other organelles are just being split, half into this side and half into this side. The final step of mitosis is telophase, and telophase is going to be when two new nuclei form around the DNA at each side, and this is also when cytokinesis occurs. Remember, mitosis is the splitting up of the DNA. Cytokinesis is the physical separation of two cells into one. So if we look here, we have two new nuclei forming. We have 4 chromosomes here, 4 chromosomes here. Now they look a little bit different than our original cell because this DNA has not been replicated yet, but they still each have 4 chromosomes and the total complement of DNA. And then our final result is 2 cells that are genetically identical to that initial cell, and we've completed the life cycle of a cell. Here's a brief video that shows actual mitosis in a cell, and you can watch this on your own with commentary, but I'm going to talk about it as well. So these little worm-like things are what you're starting to see the chromosomes condense and be visible. Here are our chromosomes, and they become visible inside the cell. They're going to align in the middle of the cell. I want you to notice here that this is not the pretty picture that you have, but the little arms are hanging out, but the main centromeres are here in the middle of the cell. Once you get to metaphase, mitosis happens pretty quickly. Here we have anaphase. Now we're going to have two new nuclei form in telophase. Cytokinesis. And now we have new cell here and a new cell here. Notice the chromosomes aren't as visible as much because the DNA is becoming uncoiled so that it can be used by the two new cells. I do want to point out there is a difference in cytokinesis between different kinds of cells, and this involves some of the basic cell differences between plants and animals. Remember that plants have a couple additional organelles, and in particular, plant cells have cell walls. And so here we have a plant cell, and because it's got this solid outer cell wall, it's not going to be able to pinch off the way a typical animal cell is. So the very dark staining here, these are the chromosomes, and both of these cells are in late telophase, so they are about to become two new cells. They're almost done with mitosis. And here we can see the cell is pinching from the outside in, and it's about to have two new nuclei. Same thing is happening here with the nuclei, but instead of pinching from the inside out, we have a small cell wall that's growing out to the sides, and this is called cell plate formation. So animals are going to pinch from the outside in, and cell walls are going to start in plants growing from the inside out. And that's mitosis and cytokinesis, taking one cell and creating two genetically identical cells.